Good morning, good afternoon, good evening all. I'm Mitchell Warren from AVAC here and delighted to welcome you to this webinar. We do have a few more people that are getting dialed out to, so we're going to start in just a minute. But you are in the right place, assuming you have dialed in either online or on the phone to a webinar with Carl Dieffenbach from the NIH, and we're going to get started in just a couple of minutes. All right, well, I think we'll go ahead and get started. This is Mitchell Warren from AVAC again, and delighted to welcome you all to this webinar that needs some introduction for some of you because it will be a new experience. I know some of you on the line undoubtedly joined in about 6 weeks ago when Carl Dieffenbach did a webinar very similar to this one, although I'll happily report that there are some new slides, I believe. Did a webinar about 6 weeks ago related to the timing right now around the recompetition of the clinical trial networks that are funded through the Division of AIDS of the National Institute of Allergy and Infectious Diseases that is part of the U.S. National Institutes of Health. And for those outside of the U.S., that's a lot of alphabet soup, and hopefully Carl's going to make that all very clear to everyone. As I said, some of you undoubtedly were on a call with Carl a while ago, and that was at a later time in the day and had a very significant proportion of U.S.-based advocates, which was fabulous. And we all agreed how important it was to do a call earlier in a day for our international colleagues to really get much of the same information and, more importantly, to engage in a conversation. And that's what we're going to do today. And I'm going to introduce it in some detail in just a moment, but let me give you a little bit of the logistics for the day. As I say, there is a presentation that Carl is going to deliver. You are all on global mute so that we can keep the line clear and be able to post a nice recording of this when we're done. But we certainly want to engage in a conversation. So I will encourage you, if you are online, you can enter questions at any point in the chat feature of ReadyTalk. You can also at any point email avac@avac.org, and we'll put those questions in the queue. And after Carl's done, I'm going to give some instruction on how to unmute your line so that you can ask questions directly to Carl. So that's kind of the run of the show. If you have logistics questions, feel free to put those in the chat feature or email them as well. Hopefully we'll get to questions and conversations in about 20 minutes, but thought it was important for Carl to outline in a brief presentation his and their thinking. And what is that thinking about? So as I said at the very outset, this is a time every 7 years, as you'll hear, where the NIH engages in thinking about the future of the HIV clinical trial networks that are funded through the NIH. I think probably everybody on the line knows that while there are many stakeholders and many funders of the HIV research enterprise, the NIH, not just in HIV, but pretty much in all biomedical research, is a disproportionately invested funder of biomedical research, and that is certainly true in the HIV space. So while any funding decision related to HIV research from any funder merits advocacy and engagement, the engagement around the NIH is obviously critical because of the enormous investment that the NIH leads. And although it's not all in the clinical trial networks, it is a major portion of the investment that Carl oversees. Carl, as many of you know, has been leading the Division of AIDS now for a number of years. And again, while it is not the only division or element of the NIH engaged in HIV HIV research, it is the largest. And we're delighted that Carl is here again with all of us to help us understand his thinking, and perhaps just as importantly, to give us an opportunity to have him understand our thinking as a community committed to advancing HIV research. And really just want to thank you, Carl, in advance because of your willingness to engage in the conversation, and not just today, but in the past and certainly over the next several weeks and months as you all think about the future. So with that, I will turn it over to Carl, and again, feel free to ask questions as we go and then at the end of the presentation.
Thank you, Mitchell. So as Mitchell has outlined for you, the purpose of today's presentation is to share with you a vision for a future for AIDS clinical research, as well as to continue a dialogue with stakeholders and community that began in January. It's important to realize that, next slide please, that as we think about a 7-year cycle, we are way through the current 7-year cycle. So to give you a sense of how seriously we take the future, it's important to realize that we are planning for events that will begin in 2020. So what we are seeking today is get a collective vision to create a forward-looking agenda so that we will have clinical trials networks and structures that will carry us through both creatively and competitively to 2027. Next slide, please. So as we think about where we have been, we think about where we were just say 10 or 15 years ago, there's been some pretty amazing successes that we as a community and as researchers can point to. We've had improvements in therapeutics. We've had developments in PrEP. We have established a number of ways of sex-specific protection. The vaginal ring from the ASPIRE trial is moving forward toward licensure. We have adult medical male circumcision. We have the results of the PROMISE study that shows that if fully implemented, treatment to pregnant women at the time of birth really has a massive reduction in transmission, mother-to-child transmission. We're building on the results of RV144. We're also looking at other forms of delivery, such as long-acting technologies for treatment and prevention. Next slide, please. NIH in 2015 hit a reset button and really refocused the priorities in AIDS research. And I think it's important that we spend a moment focusing on the overarching NIH priorities. First and foremost is HIV prevention. What we want to do is come up with methodologies to reduce HIV incidence. We want to develop the next generation of therapies, including a cure, and also develop strategies for preventing and treating the major HIV-associated comorbidities and co-infections. And from the division perspective, probably the most important one of those remains tuberculosis. And then the nature of NIH research is also to foster cross-cutting activities, including basic research, health disparities research, and above all, train the next generation of scientists. It's my hope that my generation will leave the next generation with as little residual problem from an HIV global pandemic, but if we are not 100% effective, the next generation will hopefully come in and finish the job that my generation has begun. Next slide, please. So I think it's important to have a vision of where you would like to go in the future, and I think that will then help define, if you know where you want to go, you can help, the research can then help you define the path, and then your structure can follow in a way that can help us get to this vision. So if we get to a point where we have long-acting and durable protection and treatment strategies that last for at least 6 months at a time, we can do a better job on adherence and really have a major impact on the 90-90-90 goals, as well as improve reduction in incidence. Can we get to a point where we have vaccines with efficacy of greater than 50% and be moving those vaccines either through licensure or into the long-range goal of having a vaccine that can be delivered prior to sexual debut to protect boys and girls around the world. I would like to see a point where we had cure or ART-free remission in large subsets of HIV-positive people of all ages, normalized life expectancy for all HIV-positive people, and a reduced burden in the infectious and non-infectious comorbidities, particularly tuberculosis co-infection. Next slide, please. So, in order to achieve these goals, how can we refine our clinical trials networks today and start the process? Next slide, please. So what does NIH have that's special, that's ours, that we can really have an impact on the pandemic? And formally, we are a research organization, and as such, we can work through research to develop procedures, develop new concepts that can have a profound impact on the epidemic. But ultimately, it comes from the creativity of the scientists working with community to develop biomedical innovation. Through innovation, we get new tools, new technologies that can really change the course of a disease. But additionally to the biomedical side of the house is an important component of behavioral and social science research. And one of the real strengths of NIH is being able to integrate in behavioral and social science research with biomedical innovation to give us not just an understanding of how these concepts can fit into people's lives, but also to help improve the biomedical research we do through that known behavioral understanding that we can get. Partnership is critical. Mitchell mentioned that we are one of a number of institutes at the NIH. We have very strong partnership with institutes such as the Child Health Institute, the National Institute of Mental Health, the National Institutes of Drug Abuse, the Cancer Institute, and others, and the National Institute of Heart, Lung, and Blood, that really help us extend the reach and the quality of the research that we support. But ultimately, when it comes time for implementation, there are others that are the true implementers, whether it's CDC, health departments, ministries of health around the globe, PEPFAR, that are essential partners for handoff for the research for full implementation. Next slide, please. So let's focus on biomedical innovation and behavioral and social science research for a moment. Next slide, please. But as we go forward and we're fostering this, one of the things we'd like to focus on is let's focus on three key areas of research emphasis that will help us to drive interventions and will cover all the target populations. So as we move forward, we'd like to keep our emphasis in three critical areas: methods for HIV prevention, HIV vaccines, and therapeutics. And in terms of our form following our function, what we are thinking about right now, and again, this is part of the process we're going through, is evaluating where we are and adjustments that need to be made, is a triad of clinical trials networks, one in prevention, one in vaccines, and one in therapeutics. Next slide, please. So for the next 6 or 7 slides, I'm going to talk specifically about each area, and each one will have a picture with boxes followed by a slide of words that gets into a little bit greater detail. So for HIV prevention, what we are seeking here is innovation. We need novel biomedical methods of HIV prevention, when coupled with the behavioral and social science partnerships, can really be used to address methodologies to protect the populations at greatest risk around the globe, including adolescents, young adults, and minorities, young minorities in the United States, particularly to zero in a little more detail, it's young women in sub-Saharan Africa, young black MSM, and MSM in general in the United States. We have to think about this as a youth bulge. We have to focus on this population. This is the opportunity now to intervene through research so that we can really begin to protect the next generation so that we can continue to start improving ways of drawing down the epidemic. Next slide, please. So we want to develop tools that are safe, acceptable, desired, highly effective in preventing HIV acquisition, protect the whole individual, and represent the next generation of that given strategy. And I'll pick on PEP for just a moment. You know, PEP is, in some ways, the ideal on-demand methodology to prevent HIV acquisition. But let's be frank about it. It's clunky. Who wants to take 28 days of pills if you've had an exposure? Can we get to, through innovation, a PEP that is one pill, or maybe one pill today and one pill in 2 weeks? That kind of innovation is, to my mind, relatively low-hanging fruit for collaborations between industry, community, and the National Institutes of Health. But we need to be able to think about products that fit into people's lives. Some people will want on-demand products. Some people will want products that are injectable. But at the end of the day, what we want is products that people will use. And what we see by looking in the pipeline is a need for innovation. And as such, we intend to truly maintain a very potent preclinical pipeline where the best ideas can come forward. And as those ideas reach a level of maturity, we can evaluate each to determine how and whether it's appropriate to move into clinical trials. Next slide, please. On the vaccine side, it's pretty straightforward. We need a safe, effective, and durable HIV vaccine. And as such, as we develop that, define the correlates of protection so we can improve that vaccine. And the question for the field is, what are the criteria to move a vaccine concept? Should it show a level of efficacy that we would move it forward to licensure? Next slide, please. So we really need to think about what the vision is for a vaccine and who the target population is. In a perfect world, we would have a vaccine that could be used should a Ministry of Health wish to implement it in a country that would protect youth prior to sexual debut. And that's an important thing to consider. So as such, as soon as vaccines have a reasonable safety profile, how can we initiate studies in adolescents and younger children so that we have set up the clinical data for immunology, so we can look at correlates and then define how to rapidly move these vaccines should they be licensed into youth. So that is what we mean by bridging from phase 2-3 studies to licensure and market, and partnerships with industry will be absolutely required for this, as well as ministries of health around the world. If you think about how complicated some of these vaccines are, you absolutely have to have the pharmaceutical industry there as a partner in order to even think about global scale-up for a vaccine. Next slide, please, to therapeutics. So for therapeutics, what we want is improved therapy, continue to improve therapy. If you think about where we've gone since 1995, where antiretroviral therapy was probably on the order of 20 pills a day, to now where we have fixed dose combinations that represent one pill once a day, we need to continue that level of innovation and move beyond that to something that is better, more durable, safer, and more tolerable for people who are living with HIV. We need to think about cure, and starting with an ART-free remission. And when we talk about cure, often we use that word as an abbreviation for getting to a point where we have ART-free remission. Tuberculosis remains a high priority, and complications and co-infections. Next slide, please. So here we're talking about therapy across the lifespan. Can we get to a point where we have the newer, safer, more durable antiretrovirals licensed for use in pregnancy, at birth, through childhood and adolescence? Can we get to a point where we have a cure and ART-free remission across the lifespan, irrespective of how we induce that? We also need to be able to have novel and durable prevention and treatment for tuberculosis, that also spans the lifespan. And a key part of that may be improvements in a vaccine to prevent tuberculosis. And ultimately, there are co-infections and complications in adults that are specific. For example, we have the Reprieve trial going on right now that is looking at cardiovascular disease. Long-term in infants, neuro-AIDS may be, and neurological complications may be the most important, and we wish to focus on that. And we'd also like to be able to define curative strategies for hepatitis B co-infection in adults. Next slide. So I think one of the areas that we continue to have discussion on is, can we come to an agreement within the NIH and with community about what the future is of pediatric and maternal health research? And if we can determine the what, then we can collectively determine the how, and whether it is integrated into the proposed 3 networks or has some form of a standalone leadership structure, I think can all be sorted out. But I think the important thing is, I think we have reached a good agreement here within the NIH and in talking to people that these are the top priorities in pediatric and maternal health research. And we are busy discussing and deciding the how. So clearly we want to be able to prevent sexual transmission in adolescents. We want to get HIV vaccines evaluated in pediatric populations. We want to license the newer antiretrovirals as well as the newer TB treatment and prevention strategies. We want to continue to focus on a limited subset of comorbidities in youth. And ultimately, there's also a significant amount of work that needs to be done for ART-free remission from infancy to adulthood. The ability to identify a child infected at birth, get that baby on therapy so that there is a chance that within that child's lifespan we can get to a point where they have ART-free remission or cure, is a very important part of pediatrics research as we move forward. And ultimately, we continue to need to evaluate the vaccines that are used in childhood and need to be brought forward in childhood in HIV-exposed and HIV-infected children. Clearly, there's work to be done for both, as examples, for influenza and respiratory syncytial virus. Next slide, please. So let's spend a moment talking about partnerships. Next slide, please. So a major theme of what we are seeking as we refine the networks is to continue to build partners and partnerships to make sure that the partnerships work as effectively and efficiently as possible so that we can collectively take our strengths and build a better, more effective clinical trials apparatus. So within the types of partnerships we already have that are pretty obvious to all of you are within the NIH and within the federal government. Strong partnerships with foundations such as the Gates Foundation and ultimately with the private sector. Because without the private sector manufacturing the medications and vaccines, we would not be making the progress we have. But ultimately, somebody always has to take what we discover to scale. And just like PEPFAR came along to take treatment to scale, the ability to, as quickly as possible, work with partnerships, through partnerships with implementers to facilitate handoff of these new important discoveries so that they can be brought to scale to impact incidence, to impact disease, lower the death rate, lower incidence is extremely important and a key part of what NIH is seeking through this team approach. Next slide, please. On this slide, it lays out examples of the types of partnerships we are seeking in implementation science research. Can we evaluate the appropriate combination prevention and treatment strategies so we can have the most important impact on incidence? If we think about treatment as prevention, and the HPTN 052 absolutely demonstrated that with durable virologic suppression, an individual will not transmit their virus to a sexual partner. can we figure out how to achieve durable viral suppression in adolescents? I mean, this is a group that is challenging to keep adherent. That would make a big difference as we go forward, and that's an example of the kind of partnership that we need. We need to continue, and that plays into improving initiation of therapy, improving adherence to prevention and treatment. We need to continue to partner to prevent and treat non-infectious comorbidities, and continue to think about how to perform the cross-cutting research taking us from infants to young adults. And that remains a high priority for the NIH. Next slide, please. So this is where we are on the schedule. We've been having webinars and consultations, as I said, back since January. I think it's really important that, and I appreciate AVAC's willingness to step in with 2 webinars in a relatively short time so that we can really achieve a significant amount of broadcast of our ideas, as well as then solicit feedback on what we're thinking between now and November 30th. What we intend to do is take our ideas that emerge after November 30th to our advisory committee meeting for formal approval in January, and then begin to work on our funding opportunity announcements. That's our method of soliciting the research, with a long-range goal of making awards in November of 2020. So I do realize that we are in that is years away, but it'll be here before you know it. And we have a lot of work to do collectively to continue to maintain and grow our current research agenda, to continue to discover and develop new strategies, but at the same time be ready and poised to take a big leap ahead between 2020 and 2027. So Mitchell, I think I'll pause there. It was a little longer than you probably would have liked, but for that I apologize. But now let's have some questions. And before we get to the questions, here are the email addresses and websites where you can log in and write in your comments specifically. No comment is too small. Comments, big visions, specific notes, all useful. Please feel free to contact us. And now, Mitchell, I think I'll turn it back to you for questions.
Great. Well, Dr. Dieffenbach, thank you so very much. That was terrific and not too long, lots of information to get out, and really helpful, and appreciate that. We'll come back to the slide that you're seeing right now, because we certainly want to encourage partners to be submitting both emails and going to the website. But we, before we do that, we'll want to have a conversation here. And we'll go back and remind people of that in just a little bit. As I said, a lot of questions already coming in through the chat function and through email, and thank you for those who did that, and we will be coming to many of those in just a minute. And I will also invite people to unmute their lines by pressing *7 and remute to keep the line nice and clear. I will ask that you introduce yourself and keep it relatively brief so we can keep it all going. But let me start with a few things that came in. And just to say, I know some of you were on the line before when we did the call in September with Carl, and Carl, you heard and answered a bunch of questions. There are some new people on the line asking some similar questions, so apologies in advance if some of these questions people already heard, and Carl, you've already answered, but we really do want to make sure that the people on this line can hear that. So there are a number of questions both about content in terms of research agendas and questions there, as well as about processes, and I'm going to try to put them together as best I can. There were a couple of questions, so let me focus a little bit just on the process, because you ended about the time frame and everything. And Carl, you talk about innovation, and you mentioned it several times, and a couple of people actually wrote in and said, peer review, which you have on your last slide that will happen, almost by definition, someone wrote, doesn't typically score well with innovation, because peer reviews tend to kind of look for what is already happening. And so the question really is, how will you at the NIH develop a mechanism in the review process of these proposals to really score for innovation? And related to that, someone else wrote and said, innovation is sometimes hard when you have a lot of the same investigators leading networks and leading trials year after year. So how do you encourage new leadership and new ideas, new innovation? So if you could maybe spend a little time talking about how you're going to ensure that to the degree that you can.
So let's talk about a research continuum and realize that in many ways the clinical trials network represents the ultimate crucible in which innovation is truly validated. Innovation begins first with basic research and discovery, and then that aha moment will lead to a concept that would then be developed into something that could be testable safely and effectively in people. So we have to think about innovation across the continuum. So I think that if we are going to focus on the networks, what you want is them having a pipeline of quality products, quality concepts that they would evaluate. The word innovation in clinical research gets a little complicated because you want it, maybe some innovative design issues, but you don't want it so innovative that it is unethical. And I think that we just have to remember that NIH has a remit from molecules to man. And oftentimes what we see in clinical research is we're going to follow the lead, is it doesn't take us to a phase 2, it takes us back to the bench. And we just have to keep that in mind. And so as such, we have that responsibility. The other thing I would point out is on at least the basic research side, innovation is probably one of the toughest things that reviewers score on. And I would argue that actually, if you have something that's truly innovative and can convince your colleagues that you actually know what you're talking about in a grant, those grants tend to score unbelievably well, and they tend to be the ones that get funded first.
Okay, thank you for that. Another kind of related to the process is around, someone asked, when you do these requests for proposals, is there a predetermined division of resources? So as you outlined these 3 networks, is there a prioritization or funding allocation between those 3, or how do those decisions get made in terms of, they all get 1/3 of the pie? What's the mechanism there?
So it always comes down to resources, and what you have to prioritize because we are in a level budget situation, and for every new thing we want to do, something existing must disappear. So we will continue to prioritize, and when we write the FOAs, there's a whole other component we didn't talk about today, and that's the clinical trial site where the work actually gets done. So in addition to leadership in these 3 areas, we must maintain clinical trial sites and capacity to perform the research. So it's not 3 divisions, it's actually probably closer to 4 in terms of dividing up the current resources.
And is there a predetermined sense that vaccines get, no matter what the resource envelope is there a percentage of this gets, of those 3 or 4 buckets, a predetermined percentage, or is that defined annually based on the science? So you really, really don't want to get into a situation where everything has a defined set aside. You want there to be, clearly you have to have a certain amount that is committed year after year to maintain a level of infrastructure, but you want scientific opportunity to also be a driver. So if there is something new that appears, you are flexible, you have the space to adjust and move quickly should something innovative and novel come along that you want to rapidly move forward. So yes, there is some set of predetermined quantities, but there's also flexibility from here to move things around as opportunities emerge. Great. So another word that you use several times, and people, a couple questions came in about this idea of partnership, and particularly with industry, which you mentioned. And a question came in: when industry, is there a predetermined funding requirement? So when an industry partner comes in, you said something about obviously someone has to manufacture this, and that's expertise. But when is there a pay-to-play perspective? Do partnerships require industry to be bringing their own resources, or is that the NIH networks bringing in industry by making resources available to them?
That's an interesting question, and I think that resources can be defined in a variety of ways. If a company spends $120 million making something, they've made a pretty big commitment. If they also then put resources in to help evaluate, I think the most important thing from the network perspective is an independence. That when NIH runs one of these clinical trials, for the most part, we are the sponsor, and as such then the evaluation of the concept is independent of industry in that sense. And so you're getting the best possible read on it. There's no, you're eliminating potential bias and influence. And I think that the independent, honest broker is a very important part that the American taxpayer holds us accountable for. So yes, we will, when appropriate, be asking pharmaceutical industry to put money in, but at the same time, we want to make sure that we're doing this in a way that we maintain the independence so that you as consumers of healthcare can believe that the results were generated in a fair, honest way, and you're getting the right answer that is not biased by industry influence.
Great, thanks. Speaking of influence, one of the question came in, and again, we have a lot of partners calling in from Africa this time as opposed to the previous call late in a day. But a question came in about country ownership and country leadership and country engagement from a host country perspective, and how do the networks and how does NIAID as a funder of those networks ensure that there is that degree of leadership and engagement?
That's an important question. I mean, we clearly cannot work and perform our research within a country without approval of the Ministry of Health. So well before we open a trial site or open a study, there's consultations with the ministry to make sure they understand what we're doing. Additionally, there's the IRBs and local oversight authorities that we depend on. But then should you get an outstanding result or you have something to bring forward, each Ministry of Health will need to grapple with how this fits within the budgets they have for their health budget for their nation. So in that sense, it is an ongoing dialogue and partnership that begins well before the studies ever start.
Great. Question, some now of the research agendas. You mentioned CURE. A couple questions came in. Where does CURE research fit? Is it just within the treatment network that you described in this so-called triad, and or how does this relate to the other CURE research that is funded by the NIH?
So again, we're talking. Let's remember that this represents, in principle, a pipeline where you have basic research on cells that go to animal models that ultimately would go into small starter studies in people. But when we get to a point where we have something that is ready to go to a multi-center structure, whether it's a therapeutic vaccine or broad neutralizing monoclonals plus some other agents, we'll need to make sure that we're testing it in a diversity of people and populations. And it would then be quite useful to move into research sites. Depending on the agent that is being evaluated, it may represent a partnership where a therapeutics network would, say, partner with the vaccine or prevention network because they happen to have experience with monoclonal antibodies or the particular vaccine. So that's where the magic can happen. not just at the leadership level, but also the laboratory level. We didn't talk at all about laboratories, because ultimately we're as good as the measurements we make on samples that we collect as part of the study. That will define the success or failure of our given intervention. And so one lab structure may have experience with a set of assays that are really important to another type of research, and being able to make sure that we have sharing and cross access is going to be essential.
Great. Thanks for that, Carl. I mean, there's just a couple more, then I'm going to open it up to see if anyone wants to ask on the line. Obviously, there's been a lot of conversation about what the future looks like in terms of both networks and agendas. Someone commented on the fact, your recent blog about topical and on-demand products being deprioritized. And so someone's asking here, what does that mean in the shorter term? And a couple different questions came in about the link between those existing ideas and products and a multi-purpose prevention technology agenda. Where a product might also have a contraceptive component, which is both important for health but also may help influence a user's choice of a product for contraception that also might protect against HIV. Where does that fit, both as multi-purpose products on the one hand, but also on the future truly of on-demand products?
So yeah, those are important questions. Please remember that we are halfway through this current cycle. And there's, as the questioner posed the question, they are very aware that there's ongoing significant activity in the areas of MPTs and on-demand products. And I would argue, as I look at the pipeline, and I did before I got on this call, there is not a whole heck of a lot of innovation currently in phase 1 across the spectrum. Currently we have 2 gels, a variety of rings in trial. So what we need is to complete the studies we have ongoing, look and see where we are in this agenda, see if there is something that really warrants moving forward into bigger studies, and then at the same time, then be ready to, again, follow the data. If the data says this isn't quite right or quite ready for prime time, go back to basics, go back to the bench, get a new round of innovation and bring it forward. And I think that has to be the message, is that we want something that people will use. And, maybe people will use an MPT, but, what's the data on ring use? Let's collect that through the HOPE trial, through the existing evaluations of the current round of MPTs. Let's look at it. We need to make data-driven decisions.
I would like to think everybody on the call would want that. Let me just, as a follow-up, irrespective of the structure you have, and again, would think you really reminded us that these decisions only have the implementation of new networks in 2020, so 3 years from now. So one could imagine 2 different decision paths. One, you find in HOPE and REACH that people don't really like the ring outside of an efficacy trial, because that's an unknown question, unknown answer right now. And maybe they don't like it, and therefore there isn't a future. But what happens in the 3-year cycle between now and 2020, and people really are using the product outside of an efficacy trial, much like we found with oral PrEP in, say, the PROUD study. And then you're sitting in 2020, so what's the, and again, you describe this need to think way into the future because you're making decisions that, without having that data. So, how do you manage that? So you've got, you may find that you don't pursue certain science in 2021 because people aren't using a ring, but you may find in the next 3 years that people will use it when they know it's safe and effective. And how do you pivot then to expand on that? So what's the mechanism there?
So I think that's where the partnerships come in, Mitchell. I think that if you think about what the NIH has done with oral PrEP since the completion of the IPREC study is not a whole lot. What has happened is some small implementation studies, but other others have come in to really struggle with how to implement oral PrEP. And PROUD and IPER-GAY are examples of studies where that was successful. If the ring is licensed, it really becomes a situation where how does the community, how do others, host country governments, figure out how to integrate the ring with or without contraception into their current thinking about prevention for women. And I think then the next wave of innovation isn't another ring. It's got to be something better than the ring. We're always trying to get better. So I think that this 3-year cycle, the 3-year period, really allows us to define where we are with rings. And we'll see if additional work needs to be done. Again, we will maintain a preclinical pipeline for MPTs and for discovery of on-demand agents. But again, it has to be better than what we have now because frankly, what we can compare this to is the ultimate on-demand product, which is a condom. At the same time, we also have to think about PEP as an on-demand product, and there are problems with that. And so, as I've already discussed, so I think we need to sit back and think logically about where we are going as a field and make sure that we answer the questions in the order that they need to be answered and then make decisions.
Great, thanks. Speaking of decisions, I'm going to ask one more that's come in electronically, and then maybe we'll see if people have questions on the line. A question came in, actually you talked to, and there was a slide about the future of a pediatric and maternal research agenda, and someone asked, how do you plan to ensure expertise in pediatrics and obstetrics remains within the new networks to take advantage and act on that research agenda you described if a dedicated network such as IMPACT no longer exists? And I think that's a question about impact. I think someone would also say, in the conversation similarly, how do you ensure the expertise within, say, the microbicide trials network continues even if you don't have a standalone network?
So let's focus on pediatrics for the moment, because this is where there's a lot of discussion going on here at NIH. We agree that the existing networks, whether they're PTN or the vaccine trials networks, does not have the existing expertise in these areas, because it was never in their remit. It would be incumbent at the time of network renewal that they would have to add the appropriate expertise to make sure that they could address the agenda, and that becomes a peer review type event. At the same time, NIH is still in the process of making a decision about the future of pediatric-like activity. I think that this is one of the great things about having the time now to collectively work internally to make that decision as we go forward. So, we've agreed on the research agenda. Now the focus is on how that can be most effectively implemented. But clearly we have to maintain, and everything is focused on leadership, and I think that's kind of a mistake. Because ultimately you need the clinical trial sites and the expertise at the site level also to be able to perform the appropriate studies. So I'm focused on questions related to how to achieve leadership that can generate the right studies, but at the same time maintaining a suite of places around the world where the research can actually be performed efficiently and effectively.
Great, thanks, Carl.
I say we have it. A decision has not been finalized yet on pediatrics.
Okay. I know you have a hard stop at the top of the hour, and I do want to make sure there are a lot of other questions that have come in, and I apologize I haven't gotten to them all. But let me just pause for a moment to see if anybody wants to unmute their line by hitting *7. I will ask that you introduce yourself, be brief, and *6 to go back on mute. Okay.
So why don't you just continue with the list, Mitchell? That would be most effective.
I am indeed. So there were two things. One thing you did talk about, and one thing you didn't mention in terms of HIV, things related to HIV. One, TB, and that you did talk a bit about, and one, other sexually transmitted diseases that you didn't really talk about. And someone asked a question: are those, are there clinical trial networks elsewhere in the NIH outside of DAIDS, like a TB clinical trial network or an STI clinical trial network? And if not, what's the mechanism to? Is it just on the back of HIV?
Good questions. NIAID has a number of divisions. The division of AIDS is one of them. My sister division is Division of Microbiology and Infectious Diseases, and they have a very robust STI program, including a clinical trials network that deals specifically with STIs. We are in very close contact with our colleagues for both research on TB and sexually transmitted infections with the scientists in that division. There's obviously a lot of cross talk and work that moves back and forth. So yes.
All right, and then a quick one. I think it's easy to answer. Someone wrote in after your last answer about you said a decision hadn't yet been made about whether you'd have that network. When does that decision get made? And I think you had it on your slide, but I'll just let you answer that.
So it's one of those ones I would like to. So I have another series of presentations coming up. I'd like to get this settled before November 30th, so that the community was aware, so people were not left hanging. The point is not to make this a novella where everybody has to wait till January for all these decisions, but the goal is to try to get this done in a way that is clear to the community what our intention is. We're working very closely with our colleagues and friends at the Child Health Institute to get a decision on this now.
Great. And a question that came in actually from a vaccine advocate that it could actually probably be extended to other areas as well. Question about decision making. So you have multiple efficacy trials running. How do you make those decisions? Obviously, choosing to do one more efficacy trial comes at a very high cost, as opposed to doing several smaller studies. Obviously, we need efficacy trials. So, someone asked the question: who's making those decisions? Only in the network? Is it made more broadly? And how are you planning to encourage more and better transition from early clinical trials to efficacy trials? And again, that was coming from a vaccine perspective, but I think could be applied to other areas
across
as well.
The board. Yeah, clearly there's what our basic position is your preclinical data and early immunogenicity study need to indicate that your vaccine is better than what is currently in evaluation, and significantly better. So what we have right now is a direct or a follow-up of the results from RV144 with a trial called 702, and then a new concept using some in silico designed immunogens from Janssen called 705 that is ultimately, if successful, would be a universal vaccine. So again, those are examples of how decisions were made without going into significant detail on those. But clearly we have a significant agenda preclinically that's being developed on how to trigger broad neutralizing monoclonal antibodies as a whole yet untested set of immunogens coming forward. There we're going to have to get data in early phase studies in people. Can we trigger the broad neutralizing antibodies, and if so, what does that look like? But at the same time, we have the AMP study and other studies answering the question, do monoclonal antibodies protect people from HIV acquisition? And I think that the agenda is, in that sense, well crafted to address that. But you're absolutely right. It applies across the board. To use the therapeutic analogy, quickly after we started with 3 times a day medication, everybody realized we needed to go to pills that were once a day. And many drugs that were probably really good fell out because they could not be dosed daily. Moving forward, and we think about ways of further enhancing adherence, that's where we're into sort of the longer acting agents, just because we think that the industry feels this way too. There may be better adherence with that kind of thing. So there's decisions that get made. There's input. The NIH has a series of committees that are, we have something called the Strategic Working Group, which helps make the NIH make these decisions. There's community representation on that group that weighs in and says, this is a good buy for the division. So input at all levels.
Great. And actually, you raised a question, actually two questions that relate to what's currently in research and what it would look like into the future. One you just mentioned, so someone just, as you were talking about 705, the Janssen vaccine trial, someone just asked, you say it's a universal vaccine, but as just presented actually at the recent HVTN meeting, someone just highlighted that it's only happening in a small, a region of Africa with a particular subtype. So if there's success in that trial, is there already a sense that it would, to be a truly global vaccine, that does that mean some kind of commitment that you would plan for another efficacy trial to see elsewhere? So let me get this out because I know you have short time. And separately, but similarly about a product currently in research, you talked about implementation science and the need for partnerships. Someone asked about the potential implementation of something like cabotegravir as an injectable, given the potential challenge, I think that many people recognize, of delivering a product that people need to come into the clinic 6 times a year, plus the pill taking in advance of starting an injection and then to cover a tail at the end. So the question, how do you see those playing out? Because the results of those would come out likely right around the time of this new network or these new networks.
Sure. So obviously, or not so obviously, we absolutely have to, whether Janssen chooses to run the next study on their own or there's limited partnership with the NIH is in negotiation. Clearly, the ideal would be an MSM study in the Americas. But again, this is for discussion. That's just my sense of what might make sense. So don't quote me on that. On the side of the cabotegravir, I mean, we have to realize that this is, in many ways, as you take cabotegravir, and if it shows efficacy, how can you change it? How can you improve it? Can you get to a point where there's enough of a safety signal where you don't have to start with an oral lead-in? I mean, there's all these implementation science studies that would have to be done, but clearly there would be leadership from the company that would help with that, but others would also want to step in. I could see CDC. I could see HRSA wanting to get into this. I could see ministries of health around the globe, Europeans wanting to play a role in improving this. But success will bring imitation, and that is the sincerest form of flattery, because then you can get to better long-acting or better, or maybe even oral long-acting with agents. So I think that once we have success, there will be a range of opportunities that will open up where partnerships that were not even dreamed about today will come into existence, and the research will accelerate, much like the delivery of therapy did when PEPFAR came along in 2000.
Great. I realize we are very near the top of the hour, and for those who weren't on the last call, Carl did stay on for an extra half hour or so, and that recording is available. There were even more questions. I know you have a hard stop. I will just pause for a minute. Is there any last mini question from someone? *7? All right. Carl, I know there are going to be many more opportunities. I just put back on the screen for people to be emailing and going to the website. There was a question from the last time that you answered, but I want to make it see if it's evolved at all. There was a question about seeing what the global communication and global conversation is, and I don't know if you've thought more Are you able to post summaries of the comments you're getting and how you're responding, even if people disagree? I realize you didn't ask people's permission when they submitted comments if they could be public, but have you thought about how you're going to convey what the community is talking about and how you're responding to it?
So there's a workload issue with this that I think it's not a reasonable request to have us post responses to every single question. I think if much of what has been talked about in this webinar, in previous webinars, and also then the webinars I've held with other stakeholder groups, they're all variations on the same theme. So I have, even today, most of the questions had been addressed previously, which is great that people get to hear the answers. So I would say at this point in time, I can't think of how I can possibly promise that we will post answers to all the questions. It's not practical for us to do that. We're taking in the comments. Please, everybody realize we're reading your comments. We're taking these seriously. We continue to evolve our positions. If you think about where we started, the progress on pediatrics, for example, has been a prime example of that. But please, your voice is being heard. I think that is the most important message I can leave everybody with.
Great. That is a great message to end with. I really do want to encourage people to be in touch with Carl, and you see the email and the website there. There is an enormous amount of conversation that will happen between now and November 30th. And one other thing is, I know you have to finish, Carl, but after November 30th, you talked about a lot of the internal processes, advisory committee, putting out the FOA. Will there be additional opportunities for people to engage, even if it doesn't change the structures, but in terms of ways to engage in a process, see how this unfolds, hear about peer reviews and all of that? Is there a way to stay involved?
Well, the NIH meetings are open. The advisory committee is something we broadcast, so that would be the next opportunity to see what we actually come up with. That would be my suggestion, as well as, and then when we publish the FOAs, those will be out there too. But again, we have to go into a period because of the rules around peer review and how we we work for the taxpayers of the United States. We have to do something that is fair and open. At the same time, by closing this off and going into a bubble to write these, we're making sure that we are not influenced from the outside. It's an essential component of what we do. That's why I've given this a year to take comments, because this gives us the luxury of time to evaluate the comments that we get, as well as then consider them without going through this in a rushed way. And I think we will continue up until November 30th to take in comments, to consider them. If your comment came in December 1st on World AIDS Day, would I ignore it? Absolutely not. So just, let's just get them in. We've had almost a whole year on this. Let's keep going on this path.
Great. Carl, thank you very much. Really appreciate this. For those on the line who might have missed parts of it, the recording and the slides will be up, as are the slides and recording from the call in September. There's a lot of conversation happening rightly on a lot of issues, and really want to encourage people to engage in that through the sites here. In addition, Jim Pickett and the Rectal Microbicide Advocate listserv is a wonderful place to stay engaged and connected. And I really want to just really emphasize that at least in our experience, raising these issues, raising these questions, and taking advantage of the openness, at least as Carl described on this call now, is our opportunity. So, Carl, thank you so much. I want to thank everybody for staying on the line, and I'll thank you all in advance for making sure the NIH hears what you think and want in HIV research going forward. Have great weekends, everybody, and we will stay in touch. Cheers, Carl. Thanks so much. Cheers.